So the exam will follow the same format as its, thank you. As its first two, I guess. So you have 50 questions, 40 of them will be multiple choice, regular multiple choice, A through E. Some of them will be A through D. And then the rest will be, the 10 will be true and false. And as I have, I guess, asked in the past, if anyone's kind enough to donate their notes, I'll post them to the benefit of the class, whether it's any format. So Tuesday, same, you'll have the whole time to do it. There's 4 chapters. There is a reading assignment, which I posted on Blackboard. There are practice quizzes for each. There are Jeopardy games that will help you test your vocabulary. But that's basically what the exam is going to be. It's going to be a term and a definition. There's not really many problems per se that you're going to get. It's all essentially what you see is what you get. So, and I think taking those practice quizzes strategically is the idea. So don't take them to study, take them to test what you've studied. And I cannot stress that enough, because if you use the practice quiz to give you a sense of, oh, well, this is what he's going to be asking, that's not the best idea, because I can ask something outside of these quizzes. So they should be used to test you and not to study for the test. And I can't stress that enough. So 4 chapters, 7, 8, 9, and 10. We did not discuss them in this order. We discussed them 7, 8, 10, and 9 only because 10 is molecular regulation. So I think it makes sense to discuss it after gene expression. So what I'll do is I'll cover them essentially in this order. The size of the chapter basically tells you its importance, if you will. So 10 would have the least amount of questions simply because it is the smallest. 7, 8, and 9 probably would have roughly equal numbers. But this is just to give you a big sense. You obviously have to study everything to do well. So any questions about the logistics before we start? Now the reading assignment, you probably get 2 or 3 questions from the reading, actually more from the reading assignment, because chapter 7 had that part that I highlighted yesterday at the beginning of the Word file. And this is things that you are expected to know coming into a micro course, basically having taken Bio 1, or actually even before Bio 1. But the idea, and in fact let's start with this one, the idea of DNA and RNA structure. And from all the other reading assignments which I posted, there's probably like 10 terms you need to know. There might be 1 or 2 questions from these. So these, if you don't know what I'm talking about, they are in an announcement, which I posted a few days ago. So keep that in mind. That is going to be... so as far as DNA and RNA structure, you are expected to know the difference between, purine and pyrimidine, nucleoside and nucleotide, DNA and RNA. What does 5', 3' mean? What does antiparallel mean? So for example, how does a nucleoside differ? What does a nucleoside have that a nucleotide doesn't? What does a nucleoside have that a nucleotide doesn't? And the answer is nothing, because it's the tide that has the phosphate and not the side. So you could argue the letter S, I guess. But remember, the nucleoside is smaller than the nucleotide. The nucleotide will add the phosphate to the sugar and then the base. Right? Examples of purines are adenine and guanine. Right? Examples of pyrimidines are cytosine and thymine. Now, you do not have to know the chemical structure, but you do have to know what's the difference between a purine and a pyrimidine. Right? What's the difference between a purine and a pyrimidine?
Purines have 2 rings.
Purines are bigger. They have 2 heterocyclic rings, whereas a pyrimidine doesn't. Right? Ribose in RNA, deoxyribose in DNA. Uracil in RNA, obviously, thymine in DNA. So, so for example, if you're given these, this is the percentage, and this is the percentage of the base we're talking about. And let's say you have adenine, cytosine, guanine, and thymine, and uracil. and let's say you have 20%, 20%, 20%, 0%, and then 40%. Well, whatever. And here's your key: a single-stranded DNA, single-stranded RNA, double-stranded DNA, and double-stranded RNA. Right? You look at this one. It has uracil. It doesn't have thymine. So suddenly you're looking good because you've eliminated both DNAs. It can't be this. It can't be this. What about the percentage tells you whether it's single or double? If it's single, well, if it's double, A's and U's have to be equal. C's and G's have to be equal. Since A's and U's are not equal, it has to be, single-stranded RNA. Using the same logic, if you are given that it's double-stranded molecule and asked to determine the percentage of bases, you should be able to. So for example, if the percent of cytosine, so you're given a double-stranded DNA, and you're given that the percent of cytosine is equal to 15%. What is the percent of adenine? If it is double-stranded DNA, that means cytosine and guanine have to be equal. That takes up 30%. What's left is 70 divided by 2, which has to be equal. So it's going to be 35%. Questions about this? So once you establish one, you clearly can establish. But remember, this only applies to to double-stranded DNA or RNA. It won't work if you have single-stranded. And then remember, let's link more than one chapter, if you will. True or false, origins of replication should be rich in cytosines and guanines. False, right? Because these would be more thermodynamically stable. It doesn't make sense because you need to open them up. So these tend to be rich in adenines and thymines, which are double bonded. It's not going to get more complicated than these, from that reading assignment at least. Questions about this beginning?
Sorry, can you just go over the double-stranded DNA real quick? I got it.
Right, if the percent of cytosine is 15, it should be equal to the percent of guanine, because C's and G's are equal.
Right.
So that's 30%. What's left is 70, divided by 2.
Okay.
Because they have to add up to 100.
Okay.
Because the percent of A should be equal to the percent of T, should be equal to 70 divided by 2. Okay, so DNA structure, chromosome structure, you are expected to know the formation of the chromosome. Right, loops around the central core, that's the hallmark. They have histone-like proteins. So you will get a few questions, just like I've asked in the past, which of these statements is true about the bacterial chromosome, right? It is arranged in loops around the central core. The histone, it has histones, obviously that would be false. There's histone-like proteins, etc. So you do have to know the general architecture. As far as chromosome structure, does anyone have an extra copy of the chapter notes? So that, right, any, or hard? Yeah, just to see if I'm missing something. May I? Oh, not 7? Another 7. Right, this is just to give you a... Right, and then supercoiling, you have to know the difference between type 1 and type 2, and it's very easy. Remember, type 1 cuts single-stranded, type 2 cuts 2 strands. Type 1 changes supercoils one at a time, type 2 does both. 2 strands, 2 coils at a time. And again, as far as replication goes, you do have to know the general right? So remember, in bacteria, usually you have a circular chromosome, and then you have a single origin, and then you have a single terminus. So you have bidirectional, so counterclockwise, clockwise fork. And they meet at the terminus. This is called the theta mode of replication. That's probably one point right there. One origin, one terminus, bidirectional, theta moles. So there is lots of figures, but this is just to establish. There will be at least 1 or 2 questions on the properties of polymerases. Types and properties, that is. Right? So again, you can flowchart it. We talked about DNA polymerases, right? You can, for example, talk about bacterial, eukaryotic. In bacteria, there are 3 main ones. Right? DNA pol 1, pol 2, pol 3. Here you have actually a bunch of them, but they're in Greek letters, right? Alpha, beta, gamma, delta, epsilon, whatever. And then be able to differentiate between all of these, right? This is the major replication enzyme. This is repair, this removes the primers, right? True or false? All of them polymerize 5 to 3. True. All of them remove bases 3 to 5. True, because that's proofreading. Which one is the only one that removes bases 5 to 3 going forward? Pol 1. What does it use it for? To remove the primers. So again, please know the properties. Questions so far?
Do you have to say anything about positive and negative sigma coding?
No. No, right. I mean, just again, what I just described. Right. But basically, it has to do with the direction of the superhelix. So positive is actually right. It's making it more. Negative, it's going away. So it's right. But it's, yeah, you're not going to be responsible for it. For replication, as I mentioned in class, you will get questions, whether it's transcription, replication, or translation, any one of them. You will be given proteins and asked to put them in order, right? Or put steps in order. So let's actually recap very quickly replication in general, right? They can't start, they need help. Okay? They're going to create a lot of supercoils, they need help alleviating that. And the process is fairly straightforward, right? So if you were to put proteins in order, right? And again, remember the characteristics of ODC, right? ODC is 245 base pairs. It has 4 9 mers, and it has, well, I don't know if we specified the number, so, but it has the AT-rich region, right? And these are the 13 mers, actually. They're 13 back to back to back. And this is called what? It has a special name. Right, DUE. It's called DUE. DNA unwinding element. This is, which makes sense. That's the AT-rich region. So what happens? DNA A are the first ones. They bind to the 9 mers. The DNA circles around them. AT-rich region opens. The DNA B comes along. That's the DNA B helicase. It opens it further, right? And then primase comes and puts down a primer. Now that primase is there, DNA pol 3 can come and extend. You have leading and lagging. Ultimately, you need to remove the primers. That's the role of DNA pol 1. And once you seal the primers, you need to fill the last gap after you replace it with DNA, and that's the role of DNA ligase. Remember, there's 10 true and false, so let's do an example. DNA ligase creates hydrogen bonds, true or false?
False.
False. You can't really create a hydrogen bond. Essentially, it falls into place. It creates a phosphodiester bond. Because just by putting the base there, it's a hydrogen bond already, right? There's helicase which removes hydrogen bonds. But this would be the order if you were... now, how does it terminate? Which protein is involved in termination? TUS, exactly. Now, TUS, you could argue technically can play a role even before DNA ligase. But usually this is the order. Now, you have to know what's the difference between a leading and a lagging, right? Okazaki fragments. So this probably would be as hard a question as I can ask about this. True or false, DNA pol 1 is used for both leading and lagging. DNA pol 1. True. Why would you need DNA pol 1 for the leading? To remove the priming. True or false, DNA pol 1 is required less for the leading than for the lagging. True. Because for the leading you typically have only one primer. In the lagging you have hundreds of primers, but they're both required. It's not going to go in deeper into than what I just described. So if I mentioned HU and IHF and DIA, all them proteins, all these proteins, you don't have to remember. No. So again, this is the conceptual framework. Questions about? And remember, the part that has to do with to replicate or not to replicate, you're not responsible for it. Now, even the replosome structure, it can be a little bit tedious, the one that loops in and then loops out, etc. You do not have to know the details. Again, beyond the idea of what is a replosome, right? A replosome is composed of 3 parts. What are they? Or 3 components. What's the replosome made up of? 3 basic components, which are really what you need anyway. You need helicase, you need, sorry? Polymerase, and before primase, of course. Helicase, primase, and DNA pol 3 make up the primosome. Let me just, questions about any of this? And remember, with the leading and the lagging, conceptually, let's do one real quick. Let's say you're given this replication fork. Let's say this is a labeling question. So you have this is the Y junction, right? And let's say I put a 5' here, and a 3' there, and a 5' here, and a 3' there. Right? And then you ask to determine which is the leading, which is the lagging, just from this. Right? Is this enough to tell? Right. All you know, obviously, all you need to know is 5' and 3'. That's enough to tell you leading or lagging. So how can you tell? Well, put a primer at random and see if it makes sense. Right? So let me put a primer here. So if this is 5', this is going to be what?
3'.
3'. This is going to be 5'. It's going to extend from here. That doesn't make sense. It extends from here. It can't because the replication fork is coming the other way. So this automatically is your what? Lagging. This is the leading. Why? You're going to put a 5' here. You're going to put a 3' there. And your polymerase will extend. You open, it extends. You open, it extends. Here, you're going to have to put the primer here and make an Okazaki, right? Then put someone here, or put something here, open an Okazaki, etc., etc. So this is leading, this is lagging. In fact, it's easy to tell. The leading is always the template that goes 3 to 5. Because you're going to replicate it anti-parallel 5 to 3. Okay, so that's chapter 7. Again, if I were to rank them, 7 would be the next least number of question chapter. So basically, 7 is the second smallest after 10. Questions about? Okay, 8 is a big one. So you will get a good number of questions from 8. And it is in 2 big parts, transcription and translation. And at the risk of repeating myself over and over again, you have had exposure to this material in other courses, several other courses. So what are the basics here? Same thing. You are expected, if given proteins, be able to put them in order. If given a sequence, be able to transcribe. If given an mRNA, be able to translate it. And in these cases, I would have to provide you with the genetic code if I ask you to translate something, unless it's all, methionines and tryptophans. Right? And at the risk of repeating myself again, hopefully you will get to a point where, without even thinking, someone yells a protein, you quickly say, or where it works. So, for example, EFTU. Where does it play a role? Transcription or translation? EF, elongation factor. That's automatically translation. What does EFTU do? It brings the amino acyl tRNA into the A site. This could be a question right there on the exam. So, rho factor. Where does it play a role in? Transcription or translation? Transcription, specifically termination. Right. Scheindel-Garno. Where does it play a role in? It's a sequence. Plays a role in translation. What? Initiation. That's what allows the ribosome to detect the proper reading frame, the proper start. So these are questions that hopefully, when all is said and done, you can, and again, I said that, take these notes and shuffle them. In fact, throw them, shuffle them, whatever you want, and then refill them. That's your exam. That's your exam. There's nothing more complicated. Okay, so let's start with transcription. And then be able to name the enzyme in the appropriate manner. So, for example, what is RNA-dependent RNA polymerase also called? If you have RNA-dependent RNA polymerase, it's making RNA from what? RNA, what is that? Replication. So it's RNA replicase. What is RNA-dependent DNA polymerase? It's reusing RNA to go back, reverse transcriptase. Right? What is transcriptase? DNA-dependent RNA polymerase. And what is DNA polymerase in the real sense? DNA replicase, DNA-dependent DNA polymerase. Right? So when you say DNA-dependent, it means that's the template. DNA polymerase, that's what it's making. So if it's using DNA to make DNA, that's DNA replicase. If it's using DNA to make RNA, that is transcriptase. Okay, so please know what it's just like we did for DNA polymerases. Please remember RNA polymerase, and it's easy to remember them. In fact, E. coli has only one. Eukaryotes have a division of labor, right? You have RNA pol 1, RNA pol 2, which does messenger, RNA pol 3, which does transfer, and RNA pol 4, which does organelle. But let's do transcription. So you have a sequence. Let's fill a few bases just to make it easy. So A, C, C, G, G, T, T, just to make it very easy. T, T, G, G, C, C, A. So it's a double-stranded DNA. It goes on, and you're asked to transcribe it. And you're told that RNA polymerase is going right to left. So this is the transcription route. So this is how transcription is proceeding. Which is the template strand? If transcription is going right to left, which is the transcription strand? The bottom one. How can you tell? Well, it's going to see this 3' and put the 5' base. That's what transcription does. It looks at the 3', makes a 5'. It sees an A, it puts what? It should put a T, but there's no T in RNA, there's a U, so it puts a U, it puts a U. it puts a G, it puts a G, it puts a C, it puts a C, it puts an A, it puts an A, it puts a 3'. Compare. And again, I'm showing it to you in reverse orientation because we're geared to work left to right. Well, this is your mRNA. It goes in this arrow. Compare it to this one. It's a perfect match. Well, not for the T's, of course. Right? So GG, GG, CC, CC, AA. The only difference is TT and UU. So that is why we call this the coding strand. And we call this the template strand. We call this the non-template. And we call this the non-coding. Right? So if you're given a double-stranded DNA and asked to transcribe it, all you need to do is figure out, they probably will give you enough information to figure out what is the coding. Replace the T's with U's. It has to have the same orientation, 5 to 3. Questions about... So again, please be able to differentiate between template, non-template, coding, non-coding, sense, nonsense, or anti-sense. That said, so the template strand, whether it's DNA replication or DNA transcription, always is 3 to 5. There's no exception because you have to make DNA 5 to 3, RNA 5 to 3. Beyond that, any strand, the top or the bottom, it's three-dimensional really. So any one of the two could be coding and non-coding. But for a given gene, only one of them can service the coding. So if you have 10 genes, chances are 5 of them will be quote-unquote on the top, and the other 5 will be on the bottom. So the coding could be the top or the bottom, depending on the gene. Okay, so RNA polymerase, again, be able to tell the parts. We have alpha 2. So remember, with polymerases of E. coli at least, right? RNA pol of E. coli. You have the core, and you have sigma. Together, they make the holo enzyme, right? Holo enzyme. Core, alpha 2, beta, beta prime. That's the catalytic. That's the one that adds. Potentially, you can have omega. That's omega. And sigma factor is there as a specificity factor, right? As a specificity. Because this one, again, you can do concept maps or however you want to do it. Sigma factor recognizes the promoter. And the promoter, remember, there's two. There's the -10 and the -35. This is the Tata box. And this is the Gata box, if you will. So true or false, the -10 box is transcribed. The -10 box is transcribed into mRNA.
False.
False. Because by definition, the very first base that is transcribed is what? The +1. -10 is before +1. Before is relative, but it is not transcribed. +1 is transcribed. Now, please know your directions also, at least your coding strand direction. True or false, the -10 is upstream of the +1. The -10 is upstream of the +1.
True.
True. Right? The -10 is towards the 5' end. The +1 is towards the 3' end. True. So up is 5'. Down is 3'. And remember, +1 is the first transcript. Beyond that, it is not transcribed. Upstream. Please note the sizes too. Like the normal sigma factor is sigma 70. 70 refers to the molecular weight, right? Kilodaltons. And don't worry about the other sigma factors, what they recognize. There's a table right there. So what happens in initiation, elongation, termination? Again, we can summarize it in 20 seconds. There's no need to write it. The sigma factor and the core will bind to the promoter. They will actually form a closed complex. this turns into an open complex. After about 9 nucleotides, sigma factor gets released. The core continues all the way to row-dependent or row-independent termination. How do they terminate? There's a stem loop that allows them to pause. If there is need for a row, they can't terminate on their own. If they can terminate on their own, they have a string of what after the stem loop? Not A's, actually U's, which are bonded to A's, and these are very weak, so they fall apart. It's not going to get more complicated than this. So remember, sigma factor initiation, rho factor termination, or no rho factor. But it's transcription termination. Questions about... you are not expected to know the sizes of the various RNAs, but please do know them and their function. So for example, right, well remember there's 6 of them, at least in E. coli. You have obviously a messenger RNA, you have transfer RNA, you have ribosomal RNA, you have small RNA, you have catalytic RNA, and you have transfer messenger RNA. So let's say it's an open-ended question. 1, 2, 3, 4, 5, 6. Which of these are transcribed? Which of these would be transcribed? Sorry? Actually, all of them would be transcribed. Because by definition, what is transcription? You're taking the gene and converting it into what? RNA. By definition, that's transcription. You're taking a gene, in DNA usually, and converting it into RNA. Which of these is transcribed? All of them. Which of these is replicated? All of them. Obviously, there's the gene we're talking about. Which of these is translated? Only one. This one. That's the only one that's translated. Sorry.
About 5. Can it act like transfer or
Which one? 5?
no 6?
Right, no, it is still transcribed though. There's a gene called tmRNA which transcribes it into the tmRNA.
What about transfer?
No, it plays a role in translation, but it doesn't get translated itself. Translated meaning turned into protein, and it doesn't. It helps terminate unterminated protein. Let's put it this way.
This could be a little off, but I remember hearing that they say, like a long time ago, it used to be an RNA-based world. So if the RNA can only be, brought about by being, transcribed from the DNA, how does that theory work?
Well, actually, no. There is strong evidence to suggest, and because of catalytic RNA, RNA can do the work of proteins because it can be catalytic, and it can do the role of DNA because it can hold genetic information. So there's strong evidence to suggest that it is indeed an RNA world. DNA evolved later and became so much better that essentially the RNA world was left behind. Does that make sense? Because you get the best of both worlds with the RNA. You can get the catalytic function and the storage information. The problem is it's not as efficient at both. So over time, proteins evolved to be the catalyst, and DNA evolved to be the genetic information. Right. So, and again, I'm not going to ask you, is it 50 bases or 80 bases, or, but conceptually, this is what you need to know. And very briefly, their role, right? This is translated into protein, carries the amino acids. We're going to talk about it in detail. Parts of the ribosome, we're going to talk about it in detail. Gene regulation for the most part. This plays a role in catalysis, so enzymatic, if you will, enzyme-like. And this one, its main role is to what? To Exactly, to free stuck ribosomes because the mRNA has no end stop codon. Essentially, removes stuck ribosomes. We'll go into some of the details shortly, but oh right, and then another with mRNA, there is a segment that I went over in some detail. you have to be able to differentiate between prokaryotic, right, and eukaryotic mRNA. Prokaryotic, eukaryotic. And the big difference, remember, is that prokaryotic tends to be polycistronic and relatively short-lived. This one tends to be monocistronic, meaning one gene, and long-lived, generally. With regards to each other, that is. Questions about this? Now, the genetic code, 2 or 3 questions on the code, and in fact, we can go through them real quick. You do have to know it's a triplet code. It's written in mRNA usually. If it were written in DNA, which of the 2 strands would it use? Coding, non-coding, template, non-template, whatever. The coding, it makes sense, right? Which is what? The non-template. So if you are told that AAA is lysine, what is it in the DNA code? AAA. AUG is a start codon. What is a start codon in DNA? ATG. You just replace the U's with T's because we're going to use the coding strand. We're not going to use the template. It is true or false. The genetic code is overlapping. False. Very false. There is a very false. It is this. It is commaless. It is degenerate, which means you can have more than one codon giving you the same amino acid. It is nearly universal. There are exceptions, mainly in mitochondrial DNA. And please know the start, please know the stop, and the 2 exceptions, right? UGG is the only one that codes for tryptophan, and AUG is methionine. As far as start codon, bacteria employ others. Please just know AUG. So AUG is the start, which codes for methionine. Yeah, but this could be a problem, I guess, a form of a problem. If you found, again, you isolated a new bacterium, and lo and behold, it has 5 bases. Adenine, guanine, cytosine, thymine, and bosamine. There's 5 of them. Right? It's still a triplet code, right? How many total codons would that bacterium employ?
120.
So how do you do it? How did you get to 64, which is the normal? It's 4 cubed. 4 is how many bases you have to work with, and 3 is how many that are at a time. So if you have 5 bases and still use 3, it's 5 cubed. If on the other hand you found 3 bases and it's a pentuplet code, it's 3 to the 5. Right? It's really the end rule, if you will. I want 3 bases to occur at a time. And in the classic genetic code, each position can be 4. You can have an A, a C, a G, and a T. And the second, there are also 4. And in the third, there are also 4. So if you want to start doing all the combinations that there is, it's basically 4 times 4 times 4, 4 cubed. So in my example, I have 5 bases that I can choose from. And each codon is 3. So it's 5 times 5 times 5. Right? I mean, it's essentially the end rule. Questions about the genetic codes? I think we then went over tRNA structure. Right, tRNA structure and ribosomal RNA structure. Let me discuss these real quick. With transfer RNA, you are expected to know, right, its structure. And label it. This is the anticodon, right? This is the acceptor stem. And this end is 5 or 3. And, how can you tell? This is the longer end. The longer end is always 3'. So the shorter end will be 5'. Anticodon, acceptor stem. What's the enzyme that charges it? Amino acid tRNA. That's it. It has unusual bases. You don't know. You don't have to know them. With ribosomes, I'm not going to ask you the breakdown of the 30 and the 50, but please know how they form. In other words, with bacteria, you have 30S small plus 50S large. Together, they make up 70S. In eukaryotic cytoplasm, you have 40, 60 making up 8. And do know that you have ribosomal RNA and protein, but don't know if it's 21, or is it the 5S or the 16S. So questions about this so far. Now, as far as translation goes, excuse me, just like we've talked about earlier, you do have to know the steps. You have to be able to put them in order. If given a protein, you have to know the function, right? So, for example, obviously, initiation factors play a role in initiation. Elongation factors play a role in elongation. And there's no termination factors, right? They're called release factors. Play a role in termination. And it's easy to remember, at least in E. coli, it's 3-3-3. There's 3 initiation factors, there's 3 elongation, and there's 3 termination. Okay? So you have initiation complexes, and remember the form of energy, at least the energy molecule, if you will, is GTP. It's not ATP. That is one of the few places in the cell or processes in the cell where GTP evolved to be the better, although they're equivalent in stored energy. So questions before we... Okay. Now, in class, and it's worth actually pointing if you haven't seen it, because I keep remembering to link you to it, but if you go to... well, actually, there's hundreds of them online, frankly. If you go to youtube.com/Johnny El-Rady, there's a few videos. One of them is actually called Lost in Translation. And this is a movie that several years ago my genetics class created. And we actually literally walk and dance through protein synthesis. So it might be worth your time. But, regardless, you have to be able to essentially answer questions. How do they come together? What is the role of EFTU? So let me summarize it real quick. We have 3 sites, right? We have the APE sites, or the Environmental Protection Agency, however you want to put it. But remember, the P is in the middle. Okay? So A, actually, since I'm going to use my left hand, the A, the P, and the E site, right? So A, P, and E. If you have 100 amino acid protein, how many of them enter through the A site?
99.
Sorry? 99. The only one that enters differently is the first one, which enters through the P site. In fact, since I'm going to use my right hand, so let's reverse the order. Right? So A, P, E. Still, same. Okay. Does that make sense? Okay. So the very first thing that happens is the initiator tRNA lands in the P site. What brings it? IF2. Brings it in. What does it carry? In bacteria, it carries N-formyl methionine. In eukarya and archaea, it carries methionine. So what's the next step? Essentially, after the full enzyme in full complex forms, the A site is empty. The tRNA will bring the second amino acid into the A site. What brings it? And it's actually easy to remember them. IF2 brings the start. EF2 brings the next one, but it's 2TU. Think about it. But anyway, it's the elongation factor, temperature unstable. Some of you are still thinking about it. It's TU2. Think of it this way. IF2, EF2, but TU, temperature unstable. So now they're ready to be what? Peptide bonded. Who does this, or what does this? Peptidyl transferase. And it's not a protein, it's a ribozyme. It's an RNA. So now everything is in the wrong place, right? What is, this tRNA is empty, and where is it? In the P. It's not supposed to be there. The E site is empty, it's not supposed to be empty. The A site carries what? 2 amino acids, it's not supposed to do that. So the very next step is what? Translocation, which does everything that it should do. Now the tRNA that is empty is where? In the E site. It goes away. The 2 amino acids are in the P site where they should be. And then the third one comes into the A site, brought by EFTU. Peptidyl transferase, EFG translocation. EFTU, peptidyl transferase, translocation. This goes on and on and on until what hits the A site? Stop codon. Release factors come in. They hydrolyze the bond here. This whole tRNA releases this entire entire protein. Remember, N-formyl methionine will always be on top. That's translation. If I were to draw this tRNA, and this clearly would be the amino acid chain, this end of the protein is what? This end of the protein is N or C? C, because it's N-formyl methionine. Aha. So if proteins are made N to C, how are nucleic acids made? Which directions are nucleic acids made? Sorry? How are nucleic acids made? Which direction? What are the 2 directions of a nucleic acid? 5 to 3. They rhyme. N to C, 5 to 3. So the carboxyl terminus corresponds to the 3'. The amino end corresponds to the 5'. Questions about translation? Again, I'm not going to ask you about the details of RF1, RF2, and RF3, like which one does this. There's also the ribosome release factor. As long as you know that 1-minute demo that I provided, you should be fine.